Hello, everybody. I'm Ken Davis. I'm the CEO of the Mount Sinai Health System. Most of you are familiar with our health system, but for those of you who don't, we're 8 hospitals in New York City, 400 ambulatory sites, and lots and lots of scientific grants. We're also a part of the Icahn School of Medicine at Mount Sinai, which is all part of our system. Today, I'm with my friends and faculty from Mount Sinai. From left to right is Fanny Elahi, and then we have Paul Kinney, and then Ignacio Saez. And they are all neuroscientists, but with slightly different areas of expertise. And they're going to enlighten us on why it's been so difficult to have real breakthroughs in brain disease over the last 50 years. So I'm going to start with Paul. Paul, your question is the one I just addressed. Why has it been so hard? What's going on?
That's an easy question. I would say one word really describes the problem, that's complexity. So if you think about diseases of the heart, the cardiovascular system, the liver, even cancer, I mean, we can understand those tissues and those organs, and it's that fundamental understanding that really drives new medications development, new drug discovery. When it comes to the brain, it's remarkably complex. It's the most complex known entity in the universe. And so trying to understand even the brain cells that are contained therein, how they interact, and then how that goes wrong is remarkably complex. It's not like a heart where it has one overall function, to pump blood. I'm not saying that's not easy to understand, it's complex, but the complexity really pales in comparison to the brain. And so when you try to understand the brain and how it works, and then why and when it doesn't, so you can develop new drugs, the challenge is just remarkable. And it really appears to me, and I think many people in the field would agree, that we're only now developing the technology and the understanding of how the brain works to be able to really address those challenges and identify new targets for medications development and really begin to make progress.
And you know what the irony is around that and that complexity is when I was starting in psychopharmacology, there were a couple of drugs that had all been discovered by chance. Yeah. And my mentor, who was the godfather of psychopharmacology, Leo Hollister, would say, well, we were developing this drug for an allergy, and we found that it had a brain effect. Now with science so much more sophisticated, we can't even figure that out. So my friend Fanny here is an expert in Alzheimer's disease. Tell us why we're stuck there.
I agree with Paul. And I would just add to that neurodegenerative diseases, including Alzheimer's disease, is about unwiring a system that is coming apart. And so there's a neat way through which the brain shapes up, genes, experience, shape up the brain, the functional networks. But when neurodegenerative diseases start, it's about undoing that network. And there are many different ways through which that network comes undone. And I agree that we are only now equipped with the tools to capture the complexity of that unwiring. I'm a molecular neuroscientist, so I don't think as much about structures, I think about the molecules, and we did not have the technology to measure the molecules with great precision and accuracy, and now we do. So I think the pace of progression is going to be really exponential.
Okay. And Ignacio, tell us about depression. Except for ketamine, which incidentally came from Mount Sinai for refractory depression and has been the first approved antidepressant in about 50 years, tell us why it's been so hard.
Well, the idea of complexity not only applies to the development and function of the brain, but also to its malfunctions, right? When someone has depression, not every depression case is the same. There's a wide variety of symptoms, and the constellation of symptoms that anyone in particular presents can be very different, right? So it stands to reason that a one-size-fits-all solution is never going to take care of all the needs of all our different patients, right? So the complexity also manifests itself in the variety of disease presentations that we see in our patients. And so part of the solution will be to address each disease presentation in its own individualized form. And we're going to do that through pharmacology and also through other methods that my lab uses, which address the electrical activity of the brain.
So despite that pessimistic beginning, the reality is that there are therapeutics and drugs in the pipeline. And what I'd like to now turn to is what those are. And we'll start with Ignacio and what you're doing in depression and how we may have opened a completely different therapeutic approach.
So depression is again, a complex disease, and we have multiple available treatments, including cognitive therapy and pharmacological approaches. But unfortunately, even though we have a wide array of potential therapeutic options, those don't always work. So we do need new therapeutical approaches. So what my lab does, and what we work in collaboration with other Center for Neuromodulation, Center for Advanced Therapeutics at Mount Sinai, is exploring new avenues. And the idea behind these new avenues is that the brain is an electrochemical organ, right? Neurons and brain regions communicate with one another, not just through chemicals, but also using electricity. And we've developed a variety of array of tools to address the chemical components, but not so much the electrical components. And that is also because of the complexity of the brain, right? But we're in the middle of a bit of a revolution in neurotechnology where we can actually record the electrical activity from different brain areas and try to understand how those work in different diseases and how they malfunction and so on. So one of the things that my lab is doing is we're using these surgical interventions in collaboration with colleagues in neurology and neurosurgery to implant electrodes directly into the brain of patients and deliver very anatomically accurate and targeted electrical stimulation that helps us treat the disease. So it's not a solution that is going to replace pharmacology or is not going to replace cognitive therapy, but it represents a new avenue of treatment which is in its infancy and has a ton of potential.
So let's talk about Alzheimer's disease. What do you think is in the pipeline? What can we hope for?
So much. A lot, really. I mean, I think the 2 FDA-approved drugs have just opened the door to the world believing that we can actually start treating this disease. And I would say that they're targeting end stage of the pathology where the brain is no longer clearing the proteins appropriately. And I think now we know through the effects that they've had that it's too late. So some of the medications in the pipeline at various stages of clinical trial are targeting lipid metabolism itself, cellular metabolism, inflammation, immune dysfunction, blood flow, and then my favorite, neurovascular coupling. So as the brain ages and as it starts degenerating, the communication between blood vessels and the neurons that need the nutrients, the oxygen, the signals slows down. And so that may be part of why we think a little bit slower. And as things progress, different networks of the brain become dysfunctional. So there are molecules targeting precisely that, the communication between the cells of the blood vessels and neurons.
Wow. Paul, you make many, many molecules. He's a chemist, and he makes molecules for different diseases. So tell us what you see is on the horizon and is exciting.
Well, at Mount Sinai, we take really 3 strategies to hopefully making new drugs. The first is Dr. Davis was mentioning is serendipity. We're actually looking for lightning to strike twice, which is there are known drugs out there that are natural agents. And the idea is that maybe we can take advantage of these natural agents because they act on the brain in a therapeutic manner. You mentioned ketamine. There are other drugs, psychedelic agents, for example, now where we think there's tremendous therapeutic promise. Can we understand how those drugs work and leverage that information to maybe dial out properties of the drugs we don't want and the beneficial properties of the drugs we do? So really it's about leveraging natural agents that are already out there. But on top of that, we're looking to discover new drugs based on the types of work that we do in our labs, using the most cutting-edge technologies. Instead of just having an idea this we think should be involved, it's stepping back and using the data to tell us what likely is involved. How can we intervene in those brain systems that may be dysregulated in the disease based on that information and make new drugs? And in fact, we're doing exactly that and have another number of drugs at Mount Sinai in development. And then finally, it's leveraging the body's natural system. For example, the immune system it's got tremendous potential. And it really hasn't been utilized in the context of brain disease. So that's something at Mount Sinai we're really focused on. Can we take advantage of the body's natural defense mechanism in the context of brain disorders? And I think there's tremendous potential there.
On the subject of the immune system, that connects me to Alzheimer's disease. Tell us what you think the role of the immune system and inflammation is in Alzheimer's, and is this a potential therapeutic approach?
Absolutely. I think it's very exciting because if you get to target outside of the brain You don't have the problem of delivery, and you can start doing that early. We talked about complexity of the brain. The immune system is equally complex, a network of cells that are actually mobile. So I think we need to know a lot more about the immune system to make the targets very potent. But some of the descriptions initially on brain pathology were of the microglia, the innate immune cell defense of the brain that were dysfunctional surrounding these plaques. And so I don't know, well, I know how. We had a mouse model for amyloid and we went after amyloid, but we sort of forgot for a few decades that these microglia surrounding the plaques had strange features. Now, through genetics, we're turning back, and now we're coming back to focusing on the innate immune system. But I think some really, really exciting data also suggests that the adaptive immune system could also be targeted for Alzheimer's disease and other neurodegenerative diseases.
So this is very exciting, because what it asks is, can't we get our own body to just start to eat up that amyloid and that tau and that bad stuff in Alzheimer's disease? And the reality is, it's probably happening all the time, and somehow as we get older, in some people, it just doesn't work as well. And we've got to figure out what to do to get those microglia working again. Well, let's move to those electrodes. I mean, imagine, depression's a very common disease, and our drugs, not the greatest. So do people really want to stick an electrode in their brain? I mean, doesn't that feel a little scary? So tell us, what's the downside? How do you convince somebody that they should put an electrode in their brain? Do they keep the electrode in their brain forevermore? Tell us what's going to happen.
Undoubtedly, surgery is scary. Brain surgery probably twice as scary. But these surgical interventions that I was mentioning have a long track record of being safe, and they're effective at treating specific disorders. So, for example, there's more than 200,000 patients worldwide who walk around with brain pacemakers, electrodes that are chronically implanted in a specific brain area, delivering this electricity, modulating brain activity, and improving their tremor and their motor symptoms dramatically. So it leads to a greatly improved quality of life. And so one of the fundamental ideas is that we've had this technology for a little while, and we need to find new applications for it, for other disorders where sometimes the treatments that we have are not enough. Depression being a really, really exciting example, because we are on the cusp of developing at Mount Sinai new uses for these chronically implanted electrodes in a slightly different brain region to treat these depressed patients. And these are, hopelessly depressed patients who don't respond to medication. They're basically at the end of therapeutic options. And here we have now developed this new strategy that, is very effective at treating them and giving them back their lives. So obviously it is scary, but the alternative is definitely worse.
Tell us a little more. Tell us, you've done some. Tell us what the patient response is like. How long does it take? What are the side effects? When do you pull the electrodes out? Give us some specifics.
Yeah. So obviously there's a lot of work leading up to the surgery, but basically once we're in surgery, this is a very highly collaborative effort that really needs teams of neurologists and psychiatrists and neurosurgeons and researchers. And there's a joint determination of where should we put this electrode? That's kind of a primary question. What is the area that is not working effectively? And once that's decided and we have some good ideas, we implant that electrode, and it is implanted there chronically, meaning the patient recovers from surgery, and in a day or two they go back home and they go around their lives, and they have a little battery that delivers these electrical currents and continuously modulates brain activity in a brain area that we know from previous studies is not working properly. And so we're now kind of normalizing the activity of this brain area to bring the system back into equilibrium. And so these patients do walk around with these electrodes implanted in their brains, and other than the moment of the surgery, which granted is kind of scary, they go around their lives, other than TSA at the airport, because you have some metal in your body. For the most part, they don't even notice it anymore, right? And so Yeah, and so that's how it works, and there's always the possibility to try titrate that a little bit. You can stimulate a little bit more, a little bit less, and so on and so forth. And again, this is by using this electrode in this particular brain region that's affected in depression. Where else can we put these electrodes to treat which other disorders, right?
So do they need them forevermore?
They do need it forevermore.
Even though some depression goes away with pills, you're going to leave these electrodes in for the rest of their lives.
Yeah, I mean, potentially, and this is kind of still in its infancy, but potentially you could retrain the brain and use these implants as a kind of rehabilitation rather than a neurotherapeutic, right? Where you teach the brain, okay, here's your normal state, this is what you have to get back to. And the brain is plastic, the brain can change itself, right? And it can relearn, and it can unlearn things that it's learned. So there's a possibility that we can use these as a temporary help to bring the brain activity out of a bad state and into a normal state, and at some point explant and cure the patient that way. Those are early ideas that are developing right now.
Let's turn to a different brain disease, addiction, which we're hearing and we know is such a problem. Paul, what are some of the therapeutic solutions? Can we take people at risk? Can we do something before they get addicted? I mean, how does the brain change that they become addicted? Is there a way to stop the craving with drugs? I mean, what's the future pharmacology around addiction?
My lab does a lot of work on drug addiction. For me, this is a tremendous source of frustration because unlike some of the more complex diseases of the brain where we really don't have clear insights into what the underlying drivers of the disease are, with drug addiction, we know what causes addiction. It's the agent that the person is consuming. And we have really good understanding now of how the brain is altered in response to consuming drugs or abuse. We have good understanding of the genetics. So we think we've got good targets. The problem with treatment of drug addiction in large part has been one of economics, in the sense that it wasn't really being viewed for many years as being an economically viable disease to go after. The patients were problematic financially. The clinical trials were difficult to run. What are the endpoints? And it turns out that that view now has changed over the past maybe 10 years, and major pharmaceutical companies are really interested in actually intervening, which is good considering what the US has gone through over the past 10 years with the opioid addiction epidemic. So I'm actually really optimistic about the treatment of addiction relative to other brain disorders in the near term. I think we have the targets, I think we have the capability, and now there's the willpower. And so I really think it's reasonable to suppose in the next 5 years that we'll see positive clinical trials. I mean, one example I think lots of people have probably heard about is Ozempic. This treatment of blood glucose dysregulation turns out can facilitate weight loss. Another side effect may actually be the attenuation of craving. We've known about the role of GLP, which is how Ozempic works, in its role in regulating craving for about 10 years. And so many labs, mine included, have been proposing GLP modulation for the treatment of drug addiction for quite some time. Now that you can actually see human data supporting these targets that we identify through genetics, through cell work, through animal work, actually showing positive effects in the real world, I really do think that's going to help drive next generation therapeutics for addiction.
Yep. And what's really important in what you said is that we sometimes see on TV and advertisements drugs that can block opiates. So, you took fentanyl, it was bad, here's a drug that can block the effects of that. But that's not dealing with craving. Yes. Craving is the issue. And just elaborate again. You really think you can stop craving so that even though the brain has changed and people want these drugs, that they're not going to want them anymore? They're not going to feel that craving? Tell us about it.
I mean, the brain after drug use is not the same as the brain before. It's remodeled, it's plastic. And so if you've used drugs for an extended period of time, the brain is different. And so there's no getting away from that. That's why it's a prolonged, chronically relapsing disorder. But ultimately, it's a disorder of motivation. You prioritize the drug above all other things, which means brain motivation systems have been corrupted. So the challenge for labs like mine is to understand, what does that mean? What are the brain motivation systems? And can we intervene? Not just, okay, an opioid acts at opioid receptors, let's use methadone to kind of attenuate that effect. What happens when you engage opioid signaling? How does that transmit into brain motivation systems such that you have this pathological craving that persists forever? And so what we've been looking to understand over the past 10, 20 years is, what is motivation? When does it become dysregulated? How do we intervene to dial down that motivation? And we think GLP systems are involved. There's a whole host of other neuropeptide systems that have been identified from animal studies that are now looking promising in humans, where we can intervene and say, craving, you need to dial down, and we can facilitate that process.
Great. Now, when I was doing clinical trials, there would inevitably be circumstances like this. Some patients had an amazing response to some of the drugs we were giving. And then we would do the statistical analysis on the whole group and find out that we didn't reach statistical significance, even though we knew that there were a few patients who had a miraculous recovery that couldn't have been a placebo effect. Now we recognize there's a great deal of heterogeneity in a lot of these diseases that we called Alzheimer's disease or depression or schizophrenia. So what I'd like you guys to talk about is heterogeneity. How are we going to break through so that we have clinical trials that identify people who might respond to a particular therapeutic, and we don't wind up throwing out drugs that might be good for a subpopulation, even if it's only 10%? And why don't you start?
Biomarkers.
Talk about it.
So I think conceptualization of disease had to change, and I think it's changing because we now have technology that is telling us with great precision that there are subgroups that really look alike molecularly, and that if you have memory impairment or depression or changes in your behavior, in neurodegenerative diseases, there's craving for some of the neurodegenerative disease subtypes, that we are now able to, instead of giving just a clinical diagnosis, look at the molecular abnormality, the cellular abnormalities, the contributions from the dysfunctional immune system that is attacking these neuronal networks in the brain. And that's how the page is getting turned. So the trials of the future, I think, are not going to be, do you have Alzheimer's disease, amyloid and tau in your brain? Here's a drug. It's going to be several different factors, a lot of different kinds of molecules that are going to be quantified, cell responses potentially. We talked about the immune system. We now are able to get someone's immune system stimulated in a dish and see how it's responding to various kinds of factors. All of these are going to be incorporated into deciding who gets what, when, in what dose. This is, I'd say, in the next decade. Now, in the immediate sense, we even get to predict how someone is progressing. Are they rapid progressors? Are they slow progressors? Do they have multiple pathologies? And all of these can get incorporated into making our therapeutics more precise, less toxic.
See if you can get a little more specific. Are there any drugs in the pipeline that you think, for any degenerative disease, that you think a company is going to come to you and say, we're only doing this subgroup? They are really actually using the biomarkers or using the genetics to really target some of these subgroups? Or are we still with companies that are trying it on everybody?
There are companies who are thinking about this.
Tell us, what markers are they looking at? How are they differentiating?
A lot of the companies are actually looking at the immune system markers, inflammatory markers, and some more specific subtypes of the immune system. Now, I think in general, drug companies want the largest market, but they're also realizing that their drugs are going to fail if you have this heterogeneity. So they are moving to that, and I can't be more specific than that because of confidentiality, but
Sure.
these are getting incorporated.
Paul, Ignacio, what do you think?
I would say this is exactly where Mount Sinai Health System can really play a key role. So we have patients come in, we take their basic medical status, but you collect an enormous amount of information that goes into electronic health records. Could you imagine coming in, having your health status, there's your medical record, but also then your genetics and all this information about your environment, and then doing that for 1 million people, the amount of information you would get? You can then really begin to parse people into discrete populations and say, this is really the disorder you have. It may look like someone else's disorder, but actually it's unique to you. This is the genetics that's driving it, and these are the interventions that are likely to be able to work. So when you've got just tremendous data working with artificial intelligence, that's what medicine's now going to begin to look like. And I'm really optimistic in terms of being able to parse out patient populations and really intervene in a manner that's tailored to that patient.
So we can conceptualize it, but are you seeing it happen yet? Are there companies that are coming to you with addiction and they're saying, I only want this subgroup, or I want to really look at the genome first, or I want these biomarkers first, and I'm only going to target this drug for that subgroup?
Yes, indeed.
Good.
I work with a number of drug companies. One of the drug companies actually partnered with Mount Sinai so that they can sequence the samples that they get from our patients, and then they're sharing the sequencing data with us and, of course, the patients. But then they're running those really expensive experiments for us, and then that's increasing our understanding of what's driving the disease. The reason for they're doing that is because that helps them understand how their products are going to work, where they're going to work, the patients that they work best in. Other examples are we have drug companies that come into us and say, you tell us from the genetics how we should position our product. Who's this going to work for so we can design the clinical trial properly, which was not the types of conversations we were having even 5 years ago. And so that's all changing. Companies are aware that not everyone is the same, and a disease may symptomatically look like the same as someone else, but it could be driven by completely different genetics. And so there's a real awareness of that, and that speaks to the types of interactions we're having with companies now.
Now, when you think about depression and how common it is, even though the phenotypes look the same, inevitably there must be multiple pathophysiologies that get to it. So talk to us about what you think the heterogeneity is, and how are we going to start to understand the heterogeneity of depression so that we can start to develop smart drugs?
Yeah. So, Dr. Davis, you mentioned these heterogeneity in outcomes, and the early interventions for the treatment of depression, the very first papers that came out 15, 20 years ago, they had these mixed results also, right? They had 50% of people who responded very well and 50% of people who did not respond at all, right? So it was both encouraging and a little discouraging to see that we had such a mixed response. And then we've learned to understand that this is a natural consequence of this heterogeneity, right? It's not we're coming from a pharmaceutical kind of thinking where we're going to do this thing, we're going to fix everyone, and then we realize that's not going to be the case, right? And so since we've made major improvements, and these kind of personalization and individual targeting has become the norm in surgical approaches, where every patient that comes through, they have a scanner, we look at their brain anatomy, everybody has a slightly different brain, not anybody in this room doesn't have anybody else's brain, right? So we were very careful about localizing the anatomy, the fibers that connect brain regions to one another, and about very precise localization of the electrodes, right? So once we do that, the effectiveness increases significantly, maybe not to 100%, but definitely much closer. And the future is, this is looking at a specific brain area, but now we're thinking about circuit approaches, which is not just one brain area, but multiple. And so to your point about the differences in pathology, we think that different brain areas, these malfunction differently in different subsets of depression, and we're going to be able to separate those and address them individually.
So you're going to see this with a lot of imaging studies and electrophysiology. I know that we're going to talk about this soon, but do you think there's a time in which blood or CSF is going to give us the kind of markers that will tell the physician, use this drug and not this drug?
That's a great question. I suspect that for some disorders it may, or it will, and for some others maybe not so much.
At all close? Are we doing those kind of studies? Are we getting the kind of CSF and blood markers in depression that maybe start to give us a much more narrowed, precise treatment?
Yes, we're getting there, and not just with CSF, but also with electrophysiological markers that allow you to stratify depressed patients into subtypes that might respond differently to different types of interventions.
So, Franny, you're all into blood and CSF. Tell us how you think it's going to revolutionize what's going on in degenerative disease.
And depression. What he doesn't know is that we're going to collaborate,
Sure.
and we're going to start looking at his patients. I think the brain is special because this electrical activity combined with molecules that are driving the cellular functions outside and inside the brain. And so combining the two, I think, is going to be really, really powerful. From the perspective of neurodegenerative diseases, and we've actually started doing this at Mount Sinai, because I've never seen an institution with as few silos as at Mount Sinai. People think differently about disease. You know, we all agree that the immune system is a component, and so we want to make sense of the complexity of the immune system by actually looking at the different disorders and seeing what aspects of the malfunction they share and what's specific. There will be specific aspects, right? And so inflammatory markers, I think, very nonspecific, but perhaps sensitive. You know, as the immune systems malfunction, their communication goes down, and some of these cytokines that are supposed to signal to the various cells go up. And so we can measure that. But we can measure thousands of proteins, and we can measure thousands of metabolites. And I think we were limited as to how we would actually make sense of this complexity. But as Dr. Kenny mentioned, we use artificial intelligence and machine learning, and I'm super comfortable letting the data speak to me, as opposed to having my favorite hypothesis tested in a hypothesis-driven fashion. You know, we still have hypotheses. For instance, my lab is really focused on blood vessels. We think that the interaction of the immune system and the blood vessels is a key component of neurodegenerative diseases. But exactly what molecular pathways are abnormal, it would take me decades to get there if I go one hypothesis at a time. Now we're measuring thousands of molecules and we see what is dysregulated. Then we design mechanistic studies to actually intervene on those and see whether we're actually affecting, you know, the biomarkers that I call boring, amyloid and tau. They're great readouts for efficacy.
So let me talk to you about a problem that you may be not thinking about, and probably most of the people in the audience are unaware of, but this could really affect our field. A part of the Affordable Care Act was to encourage companies to make biologics. So the deal that Big Pharma made to support the ACA was, we'll give you 12 years of market exclusivity for your biologics, no matter what the patent life is. So they all think, I'm going to make these big proteins. I'm going to make these antibodies. Here's our problem. They don't pass the blood-brain barrier. They don't get into the brain. So what happens is, suddenly neuroscience is on the decline in all these companies relative to other diseases where they have nice targets and they can make big proteins and they can charge a lot of money. Are you seeing that? And what do you think is the downstream implications of this? How much are these biologics going to be adversely affecting what is our pipeline that we need?
There's a huge impact. Most drug companies, I think, are pivoting away from small molecules, if they can, to develop biologics. And it makes sense. You have the body make a natural agent that it accepts, so the side effects are low, incredibly potent. You know, you're using your own body to heal yourself, so it makes a lot of sense. And absolutely, the role of biologics in brain disease, you know, the two don't go together, apparently, because these antibody-based biologic therapeutics don't get into the brain, apparently, which is almost certainly not the case. I don't know if anyone's read here the book Brain on Fire, where a woman had a teratoma, a type of tumor that actually makes things. And one of the things this tumor was making was an antibody against a type of receptor that's in the brain. It turned out that that receptor is well known to be involved in schizophrenia. And this individual demonstrated all the hallmarks of schizophrenia because a small amount of all it took of this antibody to penetrate into her brain to trigger what looked exactly like a psychotic state. And then once you clear the antibody, the symptoms resolve. And it turned out that that was not a unique case. In fact, there was someone in the New York Times just a few weeks ago, exactly the same case, had been in care for about 3 decades. It turned out she had an antibody against a different receptor in the brain. So this notion that antibodies don't get into the brain and they can't be utilized in a therapeutic manner for brain disorders, I think is dogma that's not going to stand up when we actually do the experiment and begin to use these things in a therapeutic manner.
Right, but as you pointed out, it was maybe only needed 1 or 2% to get into the brain to get a result, which also means 98% is floating around in the body with the possibility of adverse consequences. And it would seem to me a whole lot more efficient if instead of, by the way, we found an antibody that might get into the brain, even though we were developing it for disease X, to say, we really are going after this kind of depression, and we're going to get a pill that's going to go right into your head and be really easily absorbed and pass the blood-brain barrier. And the reason I raise this is because I'm concerned that the incentives to build long biologics and have 12 years of market exclusivity is going to kill the pipe, or at least a very grossly diminish the pipeline for these diseases. And what concerns me so much, in addition about that, is these are very common debilitating diseases. And if you look at the cost of these diseases to productivity and to the American health system, it's enormous. So the problem is we have developing these drugs to facilitate the pharmaceutical industry and their, patent protection, instead of saying, let's incentivize companies to develop drugs that we need the most because they cost the country the most money, they cost productivity the most. And what I'm thinking about is, and maybe all of you who are influential and your congressmen and your senators can talk about, there was once an Orphan Drug Act, and it incentivized companies to develop drugs for small diseases because nobody else wanted to develop them. How about if we have the flip side of that, which would be a drug incentive for the most important diseases, the most costly diseases? Why can't we incentivize companies around that? Your thoughts?
I think that'd be amazing. I mean,
But why can't we do it?
Look at the pipelines for any of the major, I won't name names, but look at any of the major pharmaceutical companies and look at our pipelines. Many of them won't even have a neuroscience pipeline. If they do have neuroscience targets, they're listed under other. Yeah. So it's not even a therapeutic area for some of the, how can that be the case when some of the most debilitating diseases we suffer from are CNS, they're brain disorders? It's incredible to me that major pharmaceutical companies don't have real effort there. I know it's hard. Many compounds have failed, but we need them, so I agree completely. That'd be amazing.
And we need to have legislation that incentivizes that. Absolutely. I think we're going to try to listen to you guys and take your questions. All right? Right away. Go ahead. Oh, we need a mic. Mics.
Hi there. I've been doing a little bit of research, not like you, of course. And my question is, are you familiar with how hormonal imbalances affect mental illness and other diseases in the brain?
We've been working a lot, actually, and we fell into it because we were interested in sex differences in terms of vulnerability to neuropsychiatric disorders. And a lot of the gene differences we saw between male and female animals turned out, as perhaps not surprisingly, but be under the control of various hormones, sex hormones, gonadal hormones. Having not worked on them before, I was just shocked by the degree to which behavior is really being controlled in a very dynamic manner by these various hormones. So we're working a lot on hormonal regulation of behavior and how we can take advantage of that from a therapeutic perspective. And actually, one of my colleagues just started a company specifically to be able to develop hormone-based therapeutics for neuropsychiatric disorders, and he's focused currently on alcohol use disorder. So I think that there's real promise there.
I think you had your hand up. Go ahead.
Yes.
Wait for the mic.
My question is there a correlation between statin use and these brain disorders?
Which one? Neurodegenerative diseases? So there's a very large national trial of actually looking at lipid-lowering, lipid-altering medications and brain outcomes, because most of the thresholds are for the heart. We actually don't know what you need to be treated with and what your threshold needs to be for brain health. So that's ongoing, and we're going to have some really good answers. And we're going to be implementing the biomarkers, hopefully, in that cohort.
But there's a corollary to what you've asked, which I'd like you to talk about, and that is so much of what becomes degenerative disease is facilitated or precipitated by other comorbidities. So maybe you can talk about things like diabetes, hypertension, and what you need to do to control those in order to avoid the bad outcomes.
Yeah. As you guys were actually talking about brain delivery, I was thinking, let's not even touch the brain, actually. Let's go to the periphery and to these metabolic imbalances. And so one of the things that we know is actually these microglia are lipid-laden. They take up the lipids. So as the lipids are levels of lipids are elevated, they alter the function of the immune system. So that's one. Inflammation is another. And we've known that for cardiovascular diseases, but now we're discovering the connections with the brain. But when we need to intervene and how much we need to intervene and what the threshold ought to be, we actually don't know. Whether you need to treat those, though, is I think, a no-brainer. And there's one clinical trial, Sprint Mind, which basically intervened on blood pressure. decreasing blood pressure under 120 as opposed to the 130 systolic blood pressure cutoff or 140, with significant decline in incidence of mild cognitive impairment and dementia. Cheapest intervention on cognition ever. And it was any medication. It wasn't a specific subclass of medication. And that also went counter to what we used to think. We thought as you age, you needed to let your blood pressure ride because your brain needed to see more, blood flow, and that was doesn't seem to be the case.
Maybe you could also talk about type 2 diabetes too, because it's a common disease. There are probably people in this group who have type 2 diabetes, and I've heard people say type 2 diabetes as it progresses is type 3 Alzheimer's disease. So tell us about that connection between diabetes and Alzheimer's disease.
Well, the advanced glycation end products, so as the levels of sugar in the blood are uncontrolled and high, they attach to the various structures of the cells and sort of cause this gradually progressive sort of silent malfunction of the cells. That's one. And they affect the brain. The other that I think is really interesting, and it's going to be an area of drug discovery, is the role of the immune system in both of those dysfunctions. And so some of the, we talked about the role of genetics, but most of Alzheimer's is actually polygenic. And this dysfunction of the immune system, or sometimes even autoimmunity, the immune system attacking itself, occurs both in diabetes and Alzheimer's, and potentially some of those therapeutics can hit both diseases. The other thing that I wanted to mention is that, again, the threshold of 6.5 of hemoglobin A1C is also, we realize, too late. So as the hemoglobin A1C or the blood sugars rise, intervention needs to occur.
Maybe I could just say one. It speaks to a bigger issue, which is I think all too often we take the brain in isolation. There's actually considerable evidence that diabetes, type 2 diabetes, may originate in the brain. It may actually be, we think of it as being a pancreatic disorder, but brain stress pathways are dysregulated, and there's considerable evidence now suggesting that it's actually the brain that's dysregulated that interacts with the pancreas. There's even evidence suggesting that some of the standard treatments for type 2 diabetes act in the brain, even though they were designed to act in the pancreas. So this notion that the brain is separate from these peripheral diseases, and these peripheral diseases have nothing to do with the brain, in many cases, that's simply not true.
Other questions? Yes, in the back. Well, I think you'll have a mic in microseconds.
I appreciate your comments, and it's all interesting to hear. I have a question for you in terms of how long this will take, and you said some of your research could take decades. With the advances in genomics right now, supercomputing and AI, which we're all hearing about today, is there a possibility that you talked about it, that your work may not take decades, or the research you want to do, which is so exhaustive, could not be done decades, but could be done in literally months? Is that a possibility in the next couple of years that will dramatically change everything you're doing? And again,
Absolutely.
equal part genomics, everything.
With my own personal experiences, it's a source of tremendous frustration. I think that things could be done much quicker and much cheaper. I think to be able to do that, though, the investment needs to be made in centers like Mount Sinai. We're not geared up to make drugs. We're geared up to identify the underlying mechanisms of disease and hopefully point drug companies in that direction. But drug companies, of course, have their own way of doing things, and it works for them. But I think we could be lean and mean and actually begin to go a bit further than we typically do in the lab, where we're like, okay, we understand this disease, we got real perspective. How about we make the drug and we push it into human clinical trials, and we move it as fast and as quickly as we can, as cheaply as we can. But typically the types of funding that labs like mine would use, it doesn't support that type of translational effort, and we can't exactly go to the medical school all the time and say, hey, could you pay for this? So it would be nice to restructure funding initiatives a little bit, and where the incentives are, that we could become lean, mean, and quick. And hopefully if we're going to fail, we fail quick. But if we're going to be successful, we benefit decades in advance of having to wait for something that may or may not make it into the clinic, depending on whether it's financially viable or not.
You see increasingly big pharma buys ideas from academia or from small startup companies. We generate targets. We figure out pathophysiology. It's another step ahead to say, can we synthesize a molecule? That's a little tougher. That takes some more money. And then the question becomes, well, if you found the molecule, are you going to test it in the animals? Are you going to show that it's safe? Does it get into the brain region? That's all drug development. What we need is a greater collaboration with Big Pharma or the NIH deciding this is going to be the place where drug discovery happens, and we got to put our money there in order to take advantage of this revolution in biology that's giving us so many more targets.
Yeah.
Yes.
Can I make a quick comment?
The man with the blue shirt here.
Can I make a quick
Oh,
comment?
please.
Yeah. So I wanted to speak on these from my standpoint, which is, electrical activity in the brain. And we've mentioned the huge potential for these single electrode interventions that we're carrying out now. Also right now, we're carrying interventions for the diagnosis of epilepsy where we don't implant an electrode, we implant dozens or hundreds of electrodes. And blink and we'll be implanting thousands of electrodes in human brains, right? And there's, concurrent with the excitement in biology and neuroscience, there's a neurotechnology boom, right? We're becoming better at creating these devices and making them biocompatible and implantable and chronic and so on and so forth. And so what used to be a trickle of data on how to use them has become a fire hose. And so together with artificial intelligence and some of these machine learning approaches, there's a real possibility for a kind of quick, maybe even exponential increase in our ability to use these effectively to treat disease. So we may be in the early stages of that, and that's reason for hope, because then maybe that could go faster.
What about early detection? Is there any studies done that shows what the cause effect of degenerative brain and what is we can do early on for a healthy brain?
That's just become possible now. I mean, for neurodegenerative diseases is what I'm going to speak to. So you can't really do early detection if your way to detect is invasive. If you require lumbar puncture to detect, no one's going to sign up for that if they're doing okay. And if you have to do PET scans equally, it's expensive. It's about $10,000 a PET scan. Again, we can't really do it at scale at a population level. But now that we can actually look in blood for these abnormalities, we are poised to do early detection, and that's exactly what we want to do at Mount Sinai. We are screening, we want to screen the population. We're doing it genetically, and we want to add the blood biomarkers to that, to people at all sorts of ages. The question about hormones, we know that they matter for the female brain. Menopause is a turning point for some people, but the studies have been done in individuals 65 and above. We've actually missed that transition of the brain into menopause, but now we can do it. I think the best design, and we're able to do that at Mount Sinai because of the incredible Sinai Million cohort and BioMe that has been created. We want to look in 20s, 30s, 40s, 50s, 60s, 70s as the brain is evolving, because you sort of need to see when the abnormality starts. That's really early detection as opposed to when someone has symptoms and comes in.
Well, there was a question in the back. Some hands in the back. Anybody, just give it somebody in the back.
Oops, okay.
I don't need a microphone. I can project my voice.
Well, somebody's got a microphone.
Okay, yeah. I was just curious as to what are some of the bioethical issues that prevent a lot of the innovations that you're making from being democratized to the public and also being cheaply accessible, because the longer it takes, obviously the more expensive it is for people who are less privileged.
Yeah.
Go ahead.
We take that incredibly seriously. I mean, data is power, and it can be used in good ways and bad ways. And I've been talking about here taking DNA samples from patients that come to Mount Sinai, combining that with health records. Talk about personal information. It doesn't get any more personal than that. So we absolutely take that incredibly seriously. So people like me, I don't work with patients. I work with cells and animals trying to understand the disease. I get access to that information completely blinded. I don't know who it comes from. I have no way of finding out where it came from, and there's no way for me to access that kind of information. And so there's firewall upon firewall upon firewall at Mount Sinai, because if we make one mistake, that's it. We will not be trusted, and this comes collapsing down. On the other hand, if we don't do this, how do we solve the big problems like brain disorders? We need the information. So we are really careful about that, and we're treading very, very lightly so that we put all the protections in place so that there's never an issue in that regard.
Last question. Go ahead. Loud.
Sleep. How does sleep factor into all of this? I mean, it's got to have
It's huge.
an impact on what you're doing.
Yeah. I would say it's kind of ignored. We spend, 1/3 of our life asleep, and yet we really have poor understanding of sleep. And yet it's something that animals do. Cells even have these cycles throughout the day. It's incredibly translational. In other words, you can measure this in an animal or cell, move it into a human, and then go back into an animal or cell. It's one of the few things that we can do reliably, and sleep almost invariably is disrupted in any kind of CNS disorder. And it's disrupted in very unique ways. So this gives us a way of really being able to get some insight into CNS disorders and move back and forth into the types of experiments that we would do in our labs to understand the biology and to come up with new drugs. It hasn't been leveraged, and it should be.
And in fact, it's one of the first things that gets disrupted in neurodegenerative diseases. And, I work on molecular detection, and I work very closely with my sleep colleagues because they are applying same thing, machine learning and AI, to wearables now in order to be able to detect at scale sleep abnormalities, fragmentation of sleep, abnormal movements in sleep for early detection. Because similar to us, where our detection was sort of expensive, speaking about the cost and generalizability, and invasive, you had to do polysomnography. You had to come and sleep not in your bed, not feel comfortable in order to have a diagnosis. And now there are really incredible algorithms that you can apply to just wearables, something you put around your wrist, that allow us to detect some abnormalities.
Well, our time is up, but we're going to hang out so you can come talk to us.